All right, let me know if you're here when I call your name, and if you're wearing red, let me know too for the extra credit. David Hodges.
Yep.
Right in front of me. All right. Sorry, I got you. Should have seen you. Eric Midkiff in red. Bryce. Cedric Richardson. Massey's here in red. Newton. Burdette. Hudson. Felder. Gotcha. Tudor is here. Asbury is here. Bowers. Hemmings. Reed. Kayla's here in reddish. Why not? Kenzie's not here. Van Cross. Anybody I missed? Well, we still got a minute. Internet's down in the room. So I guess that doesn't matter for any of you here because you're here. Well, it matters a little bit. So because the internet's down, I can't pull my PowerPoint for chapter 13 off the internet, off the Google Classroom. So we'll finish up chapter 10 today and just be done instead of jumping into chapter 13. But it might take us all day anyway because we've got to talk about all the different types of viruses. So no big deal, I guess. As a reminder, what else? So the exam's going to be sometime next week. At this point, it's almost definitely going to be a Wednesday or Friday because I want you guys to have time to study for it. And so far, the only votes I've been receiving have been for Wednesday and Friday. Make sure you do the study guides for chapters 8, 9, and 10. This weekend, I'll post the video of the exam reviews I've done previously. So you should watch that. And also, if you want to meet with me to talk about your study guide, so where we can talk about the questions you missed and the questions you got right, and talk about how I might reword the questions for the exam, let me know. I guess that's it for exam stuff. It'll be online as usual. It'll open up at 8 a.m. at the latest. But no matter what time it opens, you can start it as early as you want. And no matter what time you start it, you have to finish it and hit submit button by 8:50 as usual. So be prepared. Have your camera ready just in case something goes wrong. Like if your power goes out, your internet goes out, something like that. Or if you do it like really, like you submit it right at 8:50, go ahead and take a picture of that just as proof, just in case Google screws up and shows me that you submitted it late, have proof. What else? And also I'll be online as always. You don't need to be online, but I'll be there just in case you need me, like if something goes wrong or if you don't understand what a question is asking, just the usual stuff. So any questions about exams? All right. Any questions about anything? Let's jump into it. So again, internet's down. So I'm doing it from my computer. Let's hope it works without the battery dying. And oh yeah, that's the other reminder. As I can see, some of you might know, starting on Monday, you have to be in person or watch the video and answer the questions for attendance. So there'll be no more live streaming. You can live stream if you want, but there'll be no more credit for it. So today's the last day. Speaking of which, the word for attendance, the first word for attendance today will be agents. So if you're in person and want to get the extra credit, just send me the word agents sometime today or sometime before 9 today. And if you're online, send me a picture of an agent. Use your imagination. It doesn't have to be a non-cellular infectious agent. Just think about the word agent and send me a picture of an agent. Any questions before we jump into this? All right, here we go. Viruses and non-cellular infectious agents. This is actually quite relevant to your everyday life when you think about it, because you're not biology majors. So most likely, you're not going to be too concerned with going from DNA to RNA to protein, which is what most of this chapter was about. But sadly, you are going to be dealing with the viruses your whole life. So what is a virus? As we talked about in chapter 1, when we talked about those 8 characteristics of life, we first mentioned viruses. because they share the characteristics of living organisms in that they have genetic material in the form of nucleic acids packaged in highly organized structures. You don't necessarily need to know that, but this is just an introduction leading us into what you do need to know. So yeah, they're almost alive, right? But they're not quite alive. Why? Because of the two things right here. They're not cellular, right? And they cannot reproduce on their own. And if you want to, for independent work, you can look up the other characteristics of life from chapter 1 and compare viruses to them. Remember, if you're alive, for example, you respond to the environment. So do viruses respond to the environment? If so, how? Living things evolve, right? Populations of things evolve. Do viruses evolve? We'll go ahead and tell you that now. Yes, they do. That's important. That's why we have all these different strands of the virus that causes COVID-19, for example. Anyway, any questions about this slide? I know that's a big mess, but again, for the exam, the important thing you need to know is this second main bullet point right here. They're not alive because they're not cellular and they can't reproduce on their own. That's if I ask that. I'm more interested in you understanding how viruses work than whether they're alive or not, because honestly, that's actually a point of contention. Some scientists argue that they are, and that's another independent work topic you can look up. Make the case if you want that viruses are alive, because other people are making that case. So you can use that evidence that they've already presented and present it to me. Anyway, another way of describing viruses from your book. They're an infectious particle consisting of genes in a box. Definitely not going to ask you this. I'm just kind of describing what they are. What do I mean by genes in a box? But again, they're not cellular, so it's not like genes in a nucleus like we have. Genes in a box. That means it's nucleic acid that's wrapped up in a protein coat. And in some cases, that protein coat is also inside of an envelope with a membrane, which is a lot like a nucleus, because we didn't really get into that in chapter 4. But yeah, your nucleus is basically a membrane, right? It's in a membrane, just like your cells. And in some cases, again, just like viruses. So yes, some viruses, again, have even that in common with living things. But again, viruses can't reproduce on their own. So for that reason, they have to infect living cells and direct them the cellular machinery to make more viruses. So basically, everything you just learned about in this chapter prior to today, remember when we talked about going from DNA to RNA to protein, that whole process, viruses take advantage of that. So we have all those machines you learned about. For example, ribosomes, right? Ribosomes read the RNA and they build the protein. And we even talked about glowing pigs, right? And glowing rats, because we said this this genetic code is universal. So you can take jellyfish genes, for example, and put them in a pig, and the pig will glow. Same thing here. You can take a viral gene, put it into your cell, and your cellular machinery doesn't know any better. It reads the code and builds the proteins. And that's what we're going to talk about moving forward. Any questions about this slide? Here's a picture of a virus. Viruses look a lot different. This just happens to be one of them. But again, that just kind of shows this idea of the DNA inside of a protein code. And they will look different. You'll see all kinds of different pictures. But yeah, that's one example of one. The first one we're going to talk about is bacteriophages, or phages, however you want to pronounce it. So you can see here, before we jump into it, we're going to talk about bacteriophages, plant viruses, animal viruses, HIV, and the AIDS virus, and prions. So before we jump into this, because anybody guess what kind of organisms these bacteriophages kill? Don't overthink it. You'll probably get it. All right, we'll skip ahead. What kind of viruses do you think plant viruses kill? Plants. How about animal viruses? Good. How about bacteriophages? Got it. Yes. See, if you don't overthink it, there it is. So we can just skip this section. You already know all you need to know. Not really. We won't skip it, but basically you do know what you need to know for the exam. That is the most important thing, other than the fact that we're going to talk about the two different cycles. But let's get into it. Viruses that attack bacteria are called bacteriophages. Phage is short for, or basically stands for eaters, right? So bacteriophage means they eat bacteria. And they're always DNA viruses. So remember, we've talked about DNA, we've talked about RNA for viruses that attack bacteria. They're generally DNA viruses. I'm going to put it next to this because I'm not even going to ask you this much. You don't need to know that much detail. You don't need to know that they're DNA viruses. Just know that they kill bacteria, right? That's it from this slide anyway. And this is what they look like. To me, this always looks like some kind of space thing, like some kind of module that might attach to the outside of a space station or something, but it's not. That's what those viruses look like. So no questions about on this slide? All right. Here's an actual, instead of a drawing, an actual scanning electron microscope picture of one. You can see here there's 3 of them, and they've attached to the outside of this bacteria cell. And basically, they're like a syringe, because right in here is where they keep the genetic information. And then, like I said, they attach to the surface of the cell, and they inject the genetic information. So it literally just goes in the cell. Well, and you'll see what happens next. But that's how it works. They're like little syringes. So when I back up, you can see that picture. Imagine that as a syringe, right? So the needle comes out here, so to speak. This is where you keep the goods, specifically the DNA, right? And then it just squirts it into the bacteria. Here's another picture from your book. This one's called phage lambda. You don't even need to know that. They're just telling you what kind it is. The particular type of bacteria that it's attacking is E. coli, which again is not very important. It's just an example. And you can see here that the viral DNA is red and the bacteria DNA is green. Not in real life, but just to show you, the artist wanted to make sure you could see which DNA belonged to the virus and which DNA belonged to the bacteria. And you can see if you look closely, and we're going to talk about this, what's happened is this bacterial DNA is incorporated the viral DNA has incorporated itself into the bacteria DNA. And we're going to talk about how that happens. And here it is. Here's how we're going to talk about it happen. There's 2 different cycles. Excuse me. There's a lytic cycle and the lysogenic cycle. And before I tell you all this, basically I'll go ahead and tell you what you need to know for the exam. If anything, just remember that the lytic cycle is the short one and the lysogenic cycle is the long one. If you can remember that, then you can answer the questions about them. And the way you can remember that is that the lytic is a shorter word than lysogenic, right? Lytic has 2 syllables. Lysogenic has 4, right? So lytic is the shorter one. Lysogenic is the longer one. And again, for the exam, as far as I'm concerned, that's the most important because this is a 100 level non-biology major class, so I don't think we really need to get into too much detail about how bacteria and viruses work. How many of you are bacteria? None, right? So in a sense, this is slightly irrelevant to you. Now, don't get me wrong. If you were to go down a rabbit hole and figure out how certain other viruses work and how certain diseases work and antibacterial stuff, this is good background information to have so you're not starting at ground zero. But other than that, it's not too important, I don't think. Anyway, let's talk about it. Once the bacteria is infected, most of the times they enter what's called, again, that shorter lytic cycle, which we're going to talk about on another slide. Sometimes instead of doing the lytic cycle or in addition to the lytic cycle, they also enter the lysogenic cycle. And again, that's just the longer one, but as your book describes it, this is when the viral DNA replication happens without the phage production or the death of the cell. There's no need to write any of this down later because I'm going to explain it when I show you the picture. Basically what happens, I'll just show you the picture. It's be a lot easier. And again, if you download the PowerPoint, you can watch this little animation that shows everything happening. Same with that. This would be the lytic cycle. The next one would be the lytic cycle combined with the lysogenic cycles, and you're going to see why those 2 things are always together, or why the lysogenic cycle is always with the lytic cycle. So here's the lytic cycle. This is exactly what you might picture when you think of viruses, right? This is how it normally works. So the virus attaches itself, it inserts its DNA, right? So now here we have that viral DNA in blue. The viral DNA in green, the bacterial DNA in blue. The cellular machinery reads it. It produces the proteins, which also produce all the things that you need for the viruses. Then something assembles all those things into new viruses, and they make so many of them that eventually the cell just explodes, and then those viruses go all over the place. And then they go on to infect another bacteria virus. Make sense? That's generally how viruses work. Even animal viruses and plant viruses. Now, the nomenclature might be different, but that's generally how it works. The genetic material is inserted into the cell. The cellular machinery reads the code, does the job, builds more viruses. You build so many of them that your cell explodes with them, and then they go on to infect other cells that are nearby. Anyway, that's the lytic cycle. That's technically the short one. Any questions about that one? So again, if you're studying, remember, I'm not going to get this into that much detail. So I don't want you like reading this and saying, okay, step 1, phage attaches to cell. Step 2, the phage DNA circularizes. I didn't even mention that. So no need to worry about those details. I guess the second word for attendance will be cycle. So again, if you're in person, just want to email those words to me for extra credit, just email the word cycle. If you're online, send me a picture of a cycle. Krebs cycle, bicycle, motorcycle, I don't care. Unicycle. Anyway, if there's no questions about this, let's talk about the next one. The next one's only slightly more complicated. And this also sort of happens, so we're going to give you some examples with humans, but this sort of happens with humans too. But here's the big difference. So it's sort of the same at first, right? The phage attaches to the cell and injects the genetic material. And just like the last one, we have the bacteria DNA and the viral DNA just sitting there hanging out together. But here's where it gets different. This time, instead of that viral DNA being read and turning into proteins, what happens is it incorporates itself into the original bacterial DNA. Now, that thing's going to go through mitosis. And we've learned about mitosis, right? It makes a perfect copy. So here we go. Now we have 2 bacteria cells, each that have that viral DNA incorporated into them. And then it keeps doing that, right? Over and over. It just keeps going through mitosis. And you keep getting these cells that have the viral DNA incorporated into their one chromosome. But then eventually, so that's the lysogenic cycle, right? That's what happens. So instead of the virus killing the cell, it incorporates itself into the genome and just makes perfect copies every time it goes through mitosis. But then eventually, as I'm going to show you in the next picture, eventually the virus kicks in and then it goes back into the lytic cycle, and that's what kills it. So that's another thing to know. Sorry. So lysogenic is the longer one because when we're talking about the lysogenic cycle, we're including both of them. And lytic is the shorter cycle. Another thing to know is lysogenic, or the lytic cycle is the one that actually kills them, right? Lysogenic cycle, that's just when the virus incorporates its genetic material into the DNA. And this also happens with humans. And it's funny, your book doesn't talk that much about it, and we don't give that many details, but yes. And that's something you could look up for independent work if you want. Actually, a lot of what makes us humans is the fact that we've had viruses in our past that have been incorporated into our genome way back, because it must have been a common ancestor. We all have the same things in our genome. From one common ancestor. You could give a whole class, a whole 1 semester class on that topic. So sadly, I'll have to keep moving. Any questions about this cycle? All right, this one's going to be really quick and easy. Plant viruses. Someone already guessed it. Plant viruses attack plants. The one thing you might not know about them, and you probably don't even need to know for the exam, is they're not necessarily deadly. Instead, they stunt plant growth, which diminishes crop yields, right? So that is bad as far as humans are concerned. We don't like plant viruses because we like to eat plants, or at least feed plants to animals that we like to eat. So yeah, it's bad for that reason. It does diminish crop yields. Most of them are RNA. If I were going to ask you anything, and I probably won't, that would be the question. Excuse me. Then I might ask. Plant viruses are mostly DNA, just like bacteria viruses are mostly DNA, like we just talked about. Plant viruses are mostly RNA. Your book is the example of TMV, tobacco mosaic viruses, and it is that picture up at the top right. It's a rod shaped with a spiral arrangement of proteins. So it's kind of hard to see, but if we had time, and I'll let you just come up here and stand next to the screen and stare at it, you could see those purple things. Those are proteins just wound up in a spiral. Anyway, your book likes to talk about TMV. I'm not going to ask you any questions about it, but let's go ahead and talk about it. Obviously, it affects tobacco, hence the name, but it also affects related plants. That's a good independent work topic if you want. What are the other related plants? Anyway, it causes discolored spots on the leaves. Who cares? It's also the first virus that's ever discovered. So maybe that's why your book even mentions it, because it's the first virus that was discovered. Another good independent work topic is right. I just said it's the first virus discovered. Meanwhile, in the same chapter, when we talked about the discovery of the double helix, I went on and on about that, right? Told you about Watson and Crick and Franklin and when the paper was published and when they got the Nobel Prize. But here you just get one bullet point. This is the first virus discovered. So you can look into it if you want. You could like say, when was it discovered? Who discovered it? And if you want to really dive into it, you can write more about it if you want. Anyway, any questions about this slide? Okay. Here's a picture of what we're talking about. This is not a healthy tobacco leaf. They're not supposed to have those spots on them. We zoom into this spot. You can see this is what the whole virus looks like. And if you zoom into that chunk of the virus, you can see that that's RNA. It's kind of wound up in a coil. But then there's also protein that's wound up in a coil on the outside of it. And that's what it looks like. This is also not relevant for a 100 level non-biology major class, but we'll talk about it very quickly. Do any of you plan to go into agriculture? Okay, so no plans to anyway, so this will probably not be relevant to you, but here we go. For a virus, any plant virus to attack a virus, it has to get through the plant epidermis. So for that reason, a damaged plant is more likely to be infected. Also, sometimes insects can carry the viruses and farming tools can spread viruses. So if you were ever into agriculture, you would know if they're doing it properly. They're kind of careful about how they do things, right? Because you don't want to spread viruses. You know, if you slice one plant that has a virus and you slice another, you're likely to spread that virus. Anyway, there's also no cure for most viral plant diseases. Another good independent work topic, because the key word there is most, right? It doesn't say that there's no cure for any, just most. So which ones are there cures for? Which ones are they close to getting cures for? Which plant viruses are most important for humans? Things like that. Anyway, for all these reasons, agricultural scientists focus on prevention, and they also focus on breeding or genetically engineering varieties of crop plants that resist these viruses. Another good independent work topic, right? Which ones are they working on? Right? What plants do we have where they're trying to breed or genetically engineer them to be resistant to viruses? So any questions about this relatively unimportant slide? Unimportant as far as the exam is concerned. Obviously, we wouldn't be alive without agriculture. So for our life, this is a very important slide, but for the exam, not very important. All right, that's it for plant viruses. Pretty easy, right? Now we're going to get a little bit more complicated and talk about animal viruses. In case you didn't know, animal viruses are common causes of disease. So says your book. Now you know. Viruses cause disease in animals. None are a greater health threat than influenza, at least at the time of the writing of this book. That'd be another good independent work topic. You could compare and contrast influenza versus COVID-19. Of course, influenza has been around longer, so of course this would have killed more people. But beyond that, you could really look at which one's more deadly. And then you know that would be such a good rabbit hole to go down. How do you define deadly? Like, is it most likely to kill you if you get it, or are you most likely to get it? Things like that. Things we don't have time to talk about right now. Anyway, let's talk about the flu really quickly, even though I'm probably not going to ask you about it on the exam. Like many animal viruses, it has an outer envelope of a phospholipid membrane with spikes of protein. In other words, it's sort of like a cell in that, again, it has that outer envelope of phospholipid membrane. That's just another way of saying it has almost like a cell membrane, because that's what our cell membrane is. And yes, we also have little spikes of proteins and other things on the outside of our cells too. But theirs has spikes of proteins. And also, just so you know, that's also what the virus that causes COVID-19 is like. It has a phospholipid membrane outside with the spikes of protein on the outside as well. And just so you know, I'm definitely not going to test you on this, but this is obviously really relevant since we're technically still in the pandemic. Those spike proteins are harmless. Can't hurt you. Those spike proteins help the whole virus do its job. So without those spike proteins, yes, the virus wouldn't be as much of an issue. But the spike proteins by themselves are harmless. And that's how the mRNA vaccine works. So the mRNA vaccine, you take it, your cells read it because, it's RNA. So it reads it and builds the protein, and the protein that's building are those little spike proteins. Those little spike proteins can't hurt you, but your immune system starts to recognize them. That way, when you do get exposed to the virus, they already have your immune system should have a heads up and realize, oh, I recognize this. That's the enemy. Let's attack it. Anyway, any questions so far about the flu virus? All right. Many animal viruses also have RNA, and then they include things like flu, common cold, measles, mumps, AIDS, polio. I'm not going to ask you. That would be a good test question if this class was, more focused on health, but I'm not going to ask you that. So I'm not going to ask, all right, which animal viruses are made of RNA? Which animal viruses are made of DNA? I'm not going to ask you that, but there you go. RNA includes, again, flu, common cold, measles, mumps, AIDS, polio. DNA viruses include hepatitis, chickenpox, herpes. We're going to talk about some of these. Any questions about this side? There's a picture. This should look familiar to you. If you recognize this, that should look like a bunch of plant viruses, right? A bunch of coiled RNA inside of a coiled protein coat. Except now they're all kind of hanging out in this membrane, right? That also, again, happens to have protein spikes on the outside. All right, here we go. Here's how they work, generally speaking. This one is, let me see. Yeah, this is basically how an RNA virus works. This one's going to be a little bit more simple because it's RNA. So we're kind of skipping the middleman, right? If it was DNA, we'd have to transcribe it, then we'd have to translate it. But here, since we already have the RNA, we can skip the whole transcription part. I'm not going to ask you questions about this. So when you're studying, if you're studying the PowerPoints or you see this picture in the book, don't study this picture. Be like, I need to memorize each step. No, I'm going to talk about it, but then we're going to leave it at that. So the virus attaches itself to the outside because, again, you see how it's kind of merged in, how the yellow is merged? Because it has a membrane on the outside, just like our cells have a membrane on the outside. So they merge in, then the whole thing comes in, and the purple is protein. So it comes in, the protein kind of dissolves, if you will, and it exposes the viral RNA. So then that viral RNA goes through RNA synthesis by an enzyme that came along with the virus. So that one viral RNA is then duplicated, and you can see now that there's 2 strands of viral RNA. Excuse me. And then those two strands are used in 2 different ways. One of those strands is actually right here. It is translated, right? So that's the message, right? That's an mRNA. It's translated. Proteins are built. That's where these came from. The other strand of RNA is going to be used to be put back into the new protein or the new virus. And that's basically how it works, right? So the RNA includes information on how to build the new virus. It's also obviously the genetic copy or the genetic code that needs to be copied. So you make the proteins, you make the copy, you squish it all together, you have yourself a new virus. You build so many of them that eventually they burst the cell, which this picture isn't showing. They leave the cell, and then they go on to infect another cell. And that's basically how a viral or an RNA virus works, right there in one picture. Any questions? In a sense, you could almost think of it as translation, right? Viral, that's how they work. It's translation, no transcription, straight translation. You're just building that protein. Actually, before I move forward, whoops. Are there any questions about this slide? All right, the next word for attendance will be this one right here. I'll circle it. I'm not even going to say it. I'll just circle it. So again, if you're in person, you can just send me that word for a little bit of extra credit. If you're online, send me a picture of one of those. Again, there's all kinds of options. Doesn't have to be biological. There's all kinds of things you can take a picture of for that or find a picture of. All right, if you download the PowerPoint, you can watch this, basically what I showed you, but animated. Here's a picture of the mumps virus. It has a protein spike on the outside. You can see the membrane, the envelope also. Here we go. I'm not going to necessarily ask you this, but again, I guess it's kind of good to know. Not all animal viruses reproduce in the cytoplasm. So what I just showed you happened in the cytoplasm. So to remind you what the cytoplasm is, In the context of this conversation, there's what's inside the nucleus, right? And then there's everything else. So if we're talking about inside a cell, the cytoplasm is everything other, well, as far as this conversation is concerned, it's everything other than what's inside the nucleus. So it happens in the cytoplasm. So some examples of the herpes virus, which is also the virus that gives you chickenpox, shingles, cold sores, genital herpes. Here's a question. You don't have to answer it because of HIPAA, but I'll answer it. How many of you have had chickenpox? I've had chickenpox. And that means if you've had it, you've had the same virus that causes herpes. Also, the same virus, again, that causes shingles. I actually had shingles when I was younger, which is weird because usually it's kind of an older person thing. And I'll talk a little bit about how shingles works later. Anyway, how does this work? How does the herpes virus work? It's an enveloped DNA virus. So again, it's in that the membrane, right? The membrane envelope that I've been talking about. That part's not new. The part that is new for this conversation is that it's DNA, right? It's not RNA. So because of that, it does not reproduce in the cytoplasm. Because it's DNA, we've got to go straight to the source. So it has to get inside the cell's nucleus, which is where we keep our DNA, right? So again, I'm probably not going to ask any questions about this on the exam. But if anything, you just need to know it's DNA. And the fact that it's a DNA virus is kind of tells you about this next bullet point, which is that it reproduces in the nucleus, which makes sense because that's where we keep our DNA. Any questions about that side? All right. The herpes virus DNA can remain behind in the nuclei of certain nerve cells. I mean, any cells, but your book is going to give a little story about what happens when they remain behind in nerve cells. And when this happens, they can remain dormant until you have some sort of stress, such as an illness or emotional stress, that triggers the virus production. So again, like when you're a kid, if you ever had chickenpox, you still have the virus. It's still in you. Obviously, I don't see any of you scratching stuff. You don't look like you have chickenpox. So you might not have the symptoms of it, but the virus is in there. And if something triggers it, you could get shingles, for example. That's what happened to me. That's why I had my shingles. I think it was 22 or 23 years old. They're really young to have shingles, but the war just started. So, emotional stress triggers viral productions. Anyway, like I'm kind of getting at here, it can flare up many times throughout your life. The book points out that 75% of adults have herpes simplex 1, which is like the cold sores that you get on your mouth, and 20% have herpes simplex 2, which is genital herpes. This is not a health class, so I'm not going to ask you all that, but it's obviously kind of relevant to your life, especially if you are single. Keep that kind of stuff in mind. Anyway, any questions about this? All right. So we're done talking specifically about herpes viruses. We're going to back up and talk about animal viruses in general. This should be kind of common sense when you think about it. The damage a virus does depends partially on how quickly the immune system responds and the ability of the tissue to repair itself. Right? So if you have a virus, like back when COVID-19 was new, a lot of people, their immune system could not respond quickly. That was a new virus. So because of that, a lot of people died from it, right? The other thing is the ability of the tissue to repair itself again. So, not all tissue is created equal, which is a good independent work topic. So in your body, which tissue repairs itself quickly? Which tissues are not good at repairing themselves quickly? Which tissues cannot repair themselves at all? Anyway, on that note, your book gives this example. Usually people fully recover from colds. I hope so. I think I have one. It's either a cold or allergies. I'm not sure which. Anyway, the reason that is because respiratory tract infections, or the respiratory tract efficiently replaces damaged cells. Generally speaking, obviously there's some issues because, again, think of COVID-19. Largely a respiratory illness that killed a lot of people. And this, the cold virus, they're contrasting to poliovirus. Poliovirus is worse because it attaches nerve cells, which are not usually replaceable. So this causes permanent damage. And the only medical option to prevent poliovirus is vaccines. And it's starting to make a slow comeback. Another good independent work topic if you want to. This whole story of poliovirus. First, you can look at when was it really prevalent? What kind of what kind of damage did it do. You can look at the people like living in iron lungs because of the nerve damage. Their bodies couldn't even make them breathe, so they had to like artificially breathe. And then they came up with a vaccine. So it was almost eliminated, and now it's starting to make a small comeback because of anti-vaxxers. We're now having polio come back. It's a good time to point out, by the way, because your book doesn't, because this is not that type of class. But not everybody can take vaccines. Some people, because of their immune system or for other medical reasons, cannot take vaccines. So I'll just leave it at that. You can look it up if you want for independent work. Okay, your book always has this, The Process of Science. I always skip it because we don't have time for it. I recommend reading it because, again, it's good to think scientifically. This one happens to be about the West Nile virus, and it's relevant to us in the U.S., as you can see, because it attacked places in the U.S. Anyway, read that if you want for independent work. If you have questions about it, let me know. There we go. Let's talk about HIV and the AIDS virus. AIDS, not that I'm going to ask you, but it stands for Acquired Immunodeficiency Syndrome. This is why you're going to learn why this is such a nasty virus. Of course, by now, this book, I think it's not even that old. I think it was published in 2016. But even then, I still think a lot of this information is outdated. So everything I teach you, or everything this book teaches you, you could look it up for independent work to see if it's still relevant. This page is definitely true. AIDS, that's the disease, and it's caused by the virus HIV. Just like COVID-19 is not the virus, COVID-19 is the disease caused by the virus. Same thing here. AIDS is the disease, HIV is the virus. HIV is a retrovirus. You should know what a retrovirus is, but keep in mind it's a multiple choice question or exam. So it's not like you need to know any one of these things individually. You just need to know them all collectively and recognize that as a retrovirus. Retroviruses are RNA viruses, they're not DNA. However, even though they're RNA viruses, they reproduce with a DNA molecule. If you're thinkers, hopefully that would make you think, what the heck? That makes no sense. If it's RNA, how does it reproduce with DNA? Of course, it is 8:35 on a Friday morning, so I get it if you're not thinking yet, but let's talk about that, right? That's not right. How does this happen? It's because it's the reverse of the usual, everything I just taught you that I said you need to know for the exam. Remember I said that's going to be a question. As a matter of fact, I'll go ahead and tell you now because I've been thinking about it last night. That question is going to be worth more points than the rest of them because it's so easy and so important. So take a step back, forget about HIV for a second. The whole process goes from DNA to RNA to protein. That's all of chapter 10, right? That's what I kept teaching you. Transcription, translation. That question is going to be worth more points than the regular question. So will the question going from DNA to RNA is called what? Transcription. Going from RNA to proteins is called what? Translation. Those are going to be worth more points because that is more important than all those little details. Anyway, back to this. Retroviruses. They do the reverse of what I've been teaching you all chapter. Instead of going from DNA to RNA, it goes from RNA to DNA. And here's something I'm almost definitely going to ask you if I ask a question about this. What does that? What makes it go from RNA to DNA? Reverse transcriptase does this. And it's in the name, right? So you guys know and definitely need to know for the exam when someone's going to miss it. And again, I keep saying this, if I cried, I would do that. It's going to make me sad. Anyway, the reverse of going from DNA to RNA is called transcription. So what's the reverse of that? It's called reverse transcription. So what's the enzyme that does it? It's called reverse transcriptase. Excuse me. So many questions about basically what a retrovirus is. All right. Here's a picture of it, or, a simplified picture of it. There you've got the membrane again with the spike proteins, so that helps it get into the cell. You can see the genetic material in red on the inside of that capsule. And then you've got these little green balls. It comes with its own reverse transcriptase. So that way when the RNA, and I'll show you a picture of this, but when the RNA gets into the cell, the reverse transcriptase does its magic. It produces DNA from it, and then we'll talk about what happens after that. There it is right there. So again, hold on. Let me go forward really quickly. Let me back up. Anyway, the viral RNA comes into the cell. Again, the reverse transcriptase that came with the virus does its job. It reads the RNA and makes a strand of DNA. And then obviously we need a double strand of DNA, right? So some magic happens right there on number 2. We have a double strand of DNA that then enters our nucleus, and this is where it gets really nasty. Then it actually gets into our genome. It attaches itself in our own chromosomes. Sort of like the lysogenic stuff I was telling you about the bacteriophage, where that incorporated itself into the bacteria's genome. That's what happens here, right? It incorporates itself into our genome. So now every time we go through mitosis, we are reproducing our entire genome, including that new HIV virus that has incorporated itself into our genome. Anyway, from there, then we just have the regular process, right? We go through transcription, where that's read, and we make RNA from it. It gets its cap and tail. Maybe it goes through some splicing. It leaves, right? And what you can't see here, because we already talked about it in chapter 10, is it meets up with the ribosomes and the tRNA, and it builds the proteins. And then all of a sudden, all those parts are put together, and it builds a new virus. And then that virus leaves the cell, and it goes on to attack and kill another cell. So that's how it works. Any questions about that side? I'm not going to go into that much detail on the exam. So again, this is just one more thing. It helps to know, but you don't need to study it. There's a lot of independent work questions here, though, because your book leaves a lot out. How did we go from a single strand of DNA to a double strand? What does that? What happened to this original strand of RNA? Once it was used to make the DNA, did it just disappear? And then what is it? What puts all these parts together? So we know ribosomes are the things that are making all these proteins. But then what is it in our cells that says, all right, we need to put all these proteins together and make a virus out of it? How does that work? Independent work topics if you're interested. All right. I'll put a big LX through this just to let you know. I'm going to talk about it very quickly, but there'll be no questions about it. HIV kills white blood cells, and white blood cells are important to the immune system. And immune systems are things that, kill viruses. So also kill other things, right? The immune system keeps you healthy. So that's another reason why AIDS is such a tough disease, is it kills the very thing that's supposed to protect you from it. Also, again, like I just said, the immune system kills other things. So very often when you have AIDS, you die from something else, because AIDS just basically destroys your immune system. Then something else comes in and kills it. Anyway, at the time that this was written, there was no cure for HIV, but it was slowed by 2 categories of drugs. Before I move on, that's another good independent work topic. Is that still accurate? Is there a cure for HIV? I guess it depends on how you define that. Is it definitely something that has been shown to work? The question is, how much evidence do we have to say that yes, there's a cure, or no, it's not enough evidence yet. Anyway, these 2 categories of drugs that slow it down, they both interfere with the reproduction of the virus. I'm not going to ask you any of this, but here we go. The first type inhibits the protease enzymes, and that helps produce the HIV proteins. So again, as you know from earlier in this chapter, when you go through translation, you read the RNA and you build proteins, it reads the RNA and then you get a long strand of amino acids, right? And we call those polypeptides. And then I even asked you, I think Wednesday, I said, then what is it that helps those polypeptides fold into their shape and become a protein? So in this case, I'm telling you, at least for HIV, there's these things called protease enzymes. So yes, in this case, your cells are still producing those viral proteins, but then they need to be put together, made into their shapes. And that first type of drug helps stop that. Excuse me. And the second type of drug inhibits reverse transcriptase. So again, as you learned way back in chapter 3, when you first started learning about enzymes have inhibitors. And in this case, this particular enzyme, which does the reverse transcription, it's inhibited. And obviously because of that, you're stopping the process at the very beginning. And I'm going to give you another example of this. Here's how this works. It's a lot of form and function thing, right? This is a T. You should be familiar with this, right? A, T, C, and G. This is a particular nucleotide T. You've already learned about this one. You look here, this is what the drug looks like. And it's almost identical, right? We've got the same base. We've got a sugar, except here we have an OH. This is what you're supposed to have, but on this drug, you have an N3. And then, of course, here, oh yeah, that's it. So basically what happens is when you eat these things, when you eat this drug, and then your body's trying to use the reverse transcriptase, your body wants to put in, or your cells want to put in a T, they try to put this thing in instead. And this thing will not bond, right? Because remember, there's a sugar phosphate backbone. This won't bond. So that ends the process. That's basically how it works, but I'm not going to ask you that on the exam. Any questions about that side? Okay. The next word for attendance is not prion, but Prius. Prius is the next word for attendance, like the vehicle, the hybrid vehicle. So again, if you're in person, you could just send me the picture or the word. If you're online, send me a picture of a Prius. All right, let's talk about prions very quickly. There might be like one question about it on the exam. Prions, what are they? They're infectious proteins, and they cause brain diseases. They are misfolded forms of normal brain proteins. So that's basically what you need to know, if anything. I'm going to talk a little bit more about it, but that's basically it. They are misfolded proteins in your brain. But remember, especially with proteins, it's all about the shape, right? Proteins are, their function is based on the shape. In this case, misfolding them all obviously messes up with their function, but also in this case, it's a little bit nastier because they're infectious that way. So when one misfolded protein comes in and touches another regular protein, then it becomes misfolded. And you'll see what I mean when I show you another picture here in a second. And then they all clump together, basically putting little holes in your brain like a sponge. And obviously, if you've got little holes in your brain like a sponge, then that is going to disrupt your brain function. Not that I'm going to get into that much detail. If I ask anything about prions, again, it would be these three things right here. Infectious, misfolded proteins that cause brain diseases. Any questions about that side? Some examples of prions, and you can look up some other ones for independent work. There's something called scrappy in sheep. If you hunt in this area, you're probably already familiar with this chronic wasting disease. Mad cow disease, right? I'm sure you guys have all heard of that. Creutzfeldt-Jakob disease, that's basically, it's all the same disease. We just have a different name for it depending on what animal it's in. So Creutzfeldt-Jakob disease and Kuru, those are humans. If you want, I don't know, that's a good question. You can look that up, but I'm sure it's different for each different type of animal or species, right? Kuru is a really interesting one if you want to look that one up. That was caused by cannibalism. Or spread, I guess, So, yeah, good advice. If you're gonna be a cannibal, stick with the meat. Don't go for the brain. That'll be a test question. What kind of meat should you eat if you eat somebody? Actually, and that's it, because then it goes into this. Everything on every chapter has evolution connection. I'm always going to skip it. You should definitely read it. It's relevant. The last word for attendance will be, let's see. How about Ebola? Yeah, Ebola is the last word for attendance.
What year was that?
Which one? Oh, it's been around for a while. I remember as a kid, it was just like in the 80s. Like it comes at it'll have outbreaks and then go away, and then have outbreaks and go away. So
the one that we remember
right.
What year was that one?
I don't remember, but that is also a good independent work topic.
Before COVID.
Yeah,
That
I
was
was in fifth grade when it came back out.
Yeah.
So